I'm pleased to introduce the next speaker, Professor Rashid Bashir. He is the Distinguished Chair in Engineering and Professor of Biological Engineering and the Executive Associate Dean and Chief of Diversity at the Carle, Illinois College of Medicine at the University of Illinois at Champaign, at Urbana-Champaign. He is an NSF Early Career Award winner, and he's also the 2012 IEEE EMBS Technical Achievement Award. He serves as a co-PI on a recently funded national research traineeship for NSF, and he's the Associate Director of an NSF Science and Technology Center. He's a fellow in many organizations, including IEEE, AI, MBE, AAAS, RSC, and APS, to name a few. The title of his talk today is Micro and Nanotechnologies for Analysis of Tissue and Molecules. So please welcome him to the stage.
All right, thank you very much. I really appreciate the speakers that have given such fantastic talks all day. I'm not sure if I'm going to be able to keep up with that, but this was really, really great. And I want to really thank the organizers also for putting this great workshop together and also inviting me. So I'll give you a couple of project overview that we've been working on in our lab. We are very interested in using various types of micro and nano fabrication approaches to make devices and systems, using silicon nanofabrication, silicon MEMS, and also 3D PDMS, and also 3D biolithography, to then apply these technologies to grand challenge problems in drug screening, biorobotics, and development of hyperorgans. This work has mainly been funded by an NSF STC that I'm a co-PI in. That's with MIT, Illinois, and Georgia Tech. Roger Kam is the PI of the center, and I'm the Illinois campus lead. And then on the other half of the lab, we've been working on just, developing tools for precision and personalized medicine. And I'll really talk about a couple of examples on this side. We work on a range of technologies, including lab-on-a-chip and microfluidics, rapid point-of-care sensors, and I'll give you some examples. Also nanopores for detection of DNA methylation and sequencing. But I guess this is maybe a summary of a lot of what's been talked about, but certainly not a complete summary at all. There's been some amazing talks today, as you've seen, this promise of personalized medicine for cancer. So want to be able to maybe take, let's say, a tumor biopsy and be able to analyze it, but from a molecular analysis of the tumor, also perhaps take the tumor and expand it into microorganoids, and then be able to do some drug screening. This analysis also will inform the choices of drugs, and then there are some fantastic tools that have been described to do this personalized therapy pathway. Also, perhaps take, of course, use imaging and body fluids to then do analysis of tumors, cells, proteins, and then inform the therapy. So we've been interested in various aspects of that, and really sparked by many collaborations. I'll show you a couple of things today. I'll mainly have time to talk about the spatial mapping of nucleic acid. It's a brand new technology that we are very excited about. It's really early stage, so I'll kind of talk about that. And then also talk about our work on point-of-care sensors for detection of cells and proteins. And we have a range of collaborators from Mayo Clinic and other places, and on our campus. So let me start by this first project, which is a spatial mapping of nucleic acid. And Anurup, Akid, Julia, and Ariana have really been working on this mainly. And this is in collaboration with our colleagues at Mayo Clinic. So if you want to get a spatial map of sort of information across a tissue, that's really, really difficult. It is very important. So you have a tumor, you take a slice, and you want to look at this intratumor heterogeneity of different clones, different cells, where they're placed, perhaps, and how they're interacting. This is really challenging. Today, you do some bulk analysis, sequencing, or PCR, and you get really just sort of average information that you'll get from that entire tumor. Now, there's a lot of work, of course, on single-cell genomics and single-cell analysis that's been moving forward, but the spatial information is really something that's kind of challenging to get. So understanding the subclonal genetic diversity is very important, gives you insight into cancer pathways, and perhaps look at the different drivers of cancer progression, and also to better forecast patient outcome, again, because of this very low frequency that might be genetically distinct cells and the clones. So we need some technique to be able to really sort of unravel this mystery. So today, what is done, if you wanted to do this, you would do what's called laser capture microdissection. Take a tissue, a thin slice of the tissue, and then you would go and image different regions, and the abnormal-looking regions, you would actually cut them out using this laser and pull out this section. And then this section then is, you can go on and do sequencing or do some nucleic acid amplification. So this technique is very good sensitivity. But you need to do this manual identification of the tissue. It's very low throughput and also takes a lot of time and resources in terms of people and just being able to do this whole technique. You could also do, of course, fluorescence in situ hybridization, where you take a fluorescent probe and hybridize to a target nucleic acid. And this, again, is a very cumbersome process for those of you who have done it. It's very powerful, but takes a lot of time. You need to fix the tissue, of course, do the digestion. Denaturation takes a long time, hybridization, and this step takes a long time, and then you do the imaging. So the pluses are it is compatible with sort of single molecule resolution, but then there's a lot of noise, autofluorescence. You need this high numerical aperture confocal microscopes, and also takes a long time to actually do the whole thing from sample. If I give you a sample to result, it does take a long time. So this sort of thing came about in discussion with a colleague in Mayo Clinic. They were talking about how being able to look at certain immune cells and cancer cells in a tumor. So our approach, actually, we said, I think we can do this quite simply. So what we do is, we take this cryosection of the tissue that you cut. It's about 7 to 10 microns thick. And then rather than putting it on a glass-covered slip, which is typically done to do an immunohistochemistry or some sort of a labeling, we actually transfer it to a chip which has these wells. And by the way, we started out, so we've done this work really from a prostate cancer xenograft. It's not a human work yet. We're kind of moving forward to that next. But at this point, we get this tissue from a xenograft model. Then we place this slice on this chip and essentially have some techniques to be able to pixelate the tissue. So once we want to pixelate this tissue, we want to then fix it inside these wells. So essentially, you've taken the tissue and now cut it up into thousands of small pieces while keeping the spatial information preserved. And then we came up with a way to do loading of the reagents in a parallel way, again, in these wells, while the reagents from one well are isolated from the other well. And then we do, in this particular case, we demonstrated with a RT lamp being able to then do amplification of a specific target. So in a way, this technique is really sort of a droplet type of a droplet PCR or droplet lamp, but spatially preserved and directly from whole tissue. So we use RT lamp as our amplification reaction. We like lamp because of the fact that it is similar sensitivity as PCR, but it is more specific. It does require 4 to 6 primers. This one here shown an approach with 4 primers. So it does slightly higher cost, but it is much more sensitive and really works well from our experience from whole blood and also from directly from tissue. It is less susceptible to contaminants. It's very robust with this BST, the strand displacement polymerase. And also it's isothermal, so it doesn't require any thermal cycling. So instrumentation point of view, it's much simpler to use. So we actually designed the LAMP reagents for TOP2A. That was one of the targets that we were looking at with our collaborator for a prostate cancer. And there was really, this didn't exist in literature, so we came up with the probes for RT-LAMP for looking at TOP2A. So then I'll just show you some more details of this particular technology. So this is sort of the approach. We cyanidized that chip. We transferred the tissue. So this part right here, this is what the chip looks like. It's quite straightforward fabrication. But in this case, we use wells that were about 100 microns. And the way we etch these wells using silicon fabrication, we can get these pointed, sharp edges that are about a micron or so. And in this particular case, we had about 5,000 wells, but we've also now done it on a much larger scale. Then we want to do the tissue pixelation, fixation, and the proteinase K digestion. So this is then performed with the combination of applying a PDMS slab on top of this to pixelate the tissue with some pressure and also centrifugation. And the tissue gets actually sort of pushed inside these wells very nicely. And here you see an SEM of a dried tissue afterwards. So the tissue is all sort of maintained in here. We have fluorescent images in the paper and everything that show the cells, multiple cells. So this is about 100 microns. So you can get about 6 to 8 cells, something in that range, within one well. And this process works quite well. So this is a sort of an automated tissue microdissection that happens in about a minute across the whole tissue, versus going in and cutting out pieces with a laser in parallel. Then the next step is to actually load the reagents, and we use a capillary action-based reagent loading process. So in this particular case, again, quite a simple process that works because of the surfaces we have. So we immerse, so this whole thing, then we actually put the reagents on top. This is the lamp reagents on top of this array, and then put the whole thing in mineral oil. So what happens is that once the fluid gets inside, it stays in there because of surface tension, but then everything else is replaced by the mineral oil. So you can essentially come up with a very simple way to then isolate each well from the other, and there's really no crosstalk across the wells. So again, this step takes, over 5,000 wells, takes about a minute. With picoliter volume loading, the microinjection, if you were to do each one, would take many, many hours. And then you put it on a heating plate, and then you look at the fluorescence in real time. So this is a couple of examples I'll show you. This is one example of tissue. This is essentially the boundary right here. And then as a function of time, you can get this amplification from each pixel wherever there's high expression of that top 2A. So you get a spatial map of the expression level of top 2A at the scale of about 100 microns across the tissue. So it's very exciting. And then you can take this this fluorescence. Again, this is an exponential amplification, but you can turn this into, of course, the threshold time and get a map of the threshold time and look at the, essentially get this heat map of expression level. This is the raw data from each pixel. You can get that, and what I just want to show is the fact that you can actually get this boundaries very well isolated, that even very specific, this pixel versus that pixel, you actually get a very different signal from each pixel next to each other. So again, the tissue borders are preserved. During amplification, there's no crosstalk, and you get about 175 picoliter reactions directly from a thin tissue. Some more, because this is a new technique, I had a hard time getting it through initially because it's really hard to do controls, essentially, for this, because no direct control exists for something like this. But this was some other just control experiments with cancer versus non-cancer, which of course is easy to do. So this is a non-cancerous tissue that we sort of physically placed. We cut 2 sections and then kind of placed them next to each other. So this is the non-cancer and the cancer on the same array. And again, you don't see any amplification from the non-cancerous tissue, from the normal tissue. And again, that really works very well. So we're very excited about this. Now, there are some controls we could do that are not, again, an explicit direct one-to-one control, but kind of show that the technique really works. So one is using FTIR. So my colleague, Rohit Bhargava, he works on looking at FTIR to classify cancerous versus non-cancerous tissue. They can't tell the genetic information, but certainly can distinguish between different types of cells. So we were using a silicon well, and FTIR goes through silicon, IR goes through silicon. So you can actually use the exact same chip to do FTIR imaging. This was the same tissue, where the tissue was right around here. And then from his technique, they do this imaging and then a classification of different types of tissue. So this was the, sort of the red is the cancerous region, the white is the non-cancerous. And again, if you take the next section, so we take the next section also, and we also, in this particular case, use the exact same section, but you can see that you get a very similar sort of a map there. And then this was the region where we didn't see the amplification, which was, again, corresponding to what they found, which was the non-cancerous part. Another couple of controls, again, more details here. So this is using mRNA fish and IHC. So let's go through this kind of quickly. So this is the RT lamp as a function of time, and then turn that into the threshold. And then this is, we actually took the next section in this case, didn't take the same one, the next section, and then did a mRNA fish in collaboration with Andrew Smith. And again, you can see kind of a similar, you can just visually even see, but then if you convert this fluorescence, in this case, it's a direct hybridization-based fluorescence, then in this case, you can get the fluorescence and you can kind of compare the two. And then we also did The same thing with IHC, in this case, looking at the expression of protein, which again is the next step up in the pathway, but you can also look at that and see some similar results. Because the expression will not be, in this case, one-to-one. Not wherever you have mRNA expression, you might not get the protein expression. And then we did compare all of that. So essentially, looking at these two bar graphs right here, the yellow is where you had RT-LAMP positive and mRNA FISH positive. And then here, the yellow is the RT-LAMP positive and the IHC positive. So over 50% of the pixels were actually the same. So this turns out to be a pretty exciting approach that we think from a tissue, direct tissue, to this result, we can do this in 2 hours or less. It's really massively parallel. You can do over 5,000 independent reactions and tissue mapping. You can actually do this sequentially and build a 3D map, which we are working on now. Only requires centrifuge, sample prep, hot plate, and it's pretty simple. So really our next steps here, this was just published, so you can look at some of the details there. The next steps are to optimize it for actually FFPE tissue type. That would really make this technique very, very scalable if you can use FFPE tissue. We are working on increasing the spatial resolution down to a single cell, so you can actually get a single cell map, and also some other logical next step. And also what's really exciting, we're working on this, maybe I can come back and talk about this someday, is this technique we think is actually directly applicable to doing a single cell RNA-seq with a spatial information. So we're actually working on that next. Okay, in the remainder of the time, I'll talk about a couple of things really quickly also. So let me talk about our work on point-of-care detection of cells and protein. This is very applicable, we believe, to cancer. Some of it we have applied, and some of it we've also been working on other applications, such as sepsis and infectious disease. But what we wanted to do was to really sort of take the power of a hematology analyzer, which does a complete blood cell count, and a flow cytometer, which can detect proteins and cell surface receptors, and combine that into one. Now, of course, as mentioned earlier, we're not claiming to do everything under the sun like Theranos did, so this is not what that technology is, but I do believe that a small subset of all the parameters can be detected reliably at a point of care. And there's been many examples, great examples that you heard today, but our goal was really to come up with a technology which can detect cells and proteins and DNA at the same time in the same platform, if we can, in the exact same platform. That's probably what distinguishes it from other approaches. So our approach is based on something like this, where we take whole blood, and we can, in this case, we lyse the red blood cells in a microfluidic mixer. So outcome of this region are the white blood cells. Then you have to quench the lysing, otherwise the lysing buffer continues to also damage the white blood cells. So we then quench the lysing, collect the white blood cells, and then we use a standard Coulter counter-like of an approach to count the cells. And also at that point, you can interrogate them to differentiate between the lymphocytes versus the granulocytes, monocytes. And then we have a region, a depletion region, we call it, a region where antibodies are attached to capture specific cells. And then we count what's going out. So the difference of these two then tell us what's been captured. So this becomes an electrical way of telling what's been captured, which cells have been captured, and you can also multiplex and scale this. So we demonstrated this for a couple different cell types. This is kind of what an earlier version of the chip looked like. And what's really exciting is that from the same drop of blood, then we can do WBC neutrophils, granulocyte monocytes together, and then we have shown these cell types, and then also more recently, protein biomarkers from the same drop of blood at the same time. Let me just see if this thing will work here. So this is whole blood coming into this chip. At this inlet, there is lysing solution coming in from the sides. This is the bloodstream. And a couple of these turns down, the red blood cells get lysed. The solution essentially gets transparent, which is with just the white blood cells remaining, which are 1,000 times less. So the solution, so about a 10 microliter of whole blood is what's used, and we end up at here about 120 microliters of blood, essentially, with all the lysing and the quenching reagents. And then that goes through this capture region, so it gets counted, captured, and then counted. So let me show you a couple of these things. So the capture here, this is the approach pioneered by Mehmet Toner and others, but we use something very similar where we have posts to then capture cells with antibodies attached. We can capture, here is shown CD4 and CD8 T cells. with a very high capture efficiency of over 85 to 90% at about a 20 microliter of flow rate. So that takes about 5 minutes to flow that entire solution of about 100 to 120 microliters of fluid. And then you can count. These are the counts, pulses of single cells, and you can take the data from your counters. This is counter 1, counter 2, and the difference of these two lymphocytes in this case would tell you what's been captured if the antibodies were for specific lymphocytes. So we did this for HIV, actually, and this shows the data for flow cytometry control in the x-axis, and the y-axis is from our chip. You get very good correlation for CD4 and CD8 for healthy donors, and also this is for HIV-infected patients. Then more recently, we also have demonstrated this for CD64 neutrophils. And again, this approach also captures cells based on expression level. So you actually can get, this is the controlled normalized expression level from a flow cytometer from 60 patient samples from the hospital. And then the y-axis is the data from our chip, and we get very good correlations. So I'm showing this as demonstration, and then the idea is in this case is that clearly this is not as low as what a CTC concentrations are, but this would be for other cell types, looking at immune cell types or other types of cells with specific receptors that you want to capture on chip at a point of care. In this case, we're looking at a CD64 it's a neutrophil, so it's this difference of the second peak, which is the granulocyte monocyte peak, and the difference of these two peaks is what tells us this specific CD64 capture. Now, what's also very exciting is that using the exact same platform, we can use beads and nanoparticles to capture proteins. So if you have, what I'm not showing here is the capture of let's say, proteins or potentially even DNA, which is what we're working on next. If you capture those from whole blood, and then those particles are then flown, then essentially you can tag another secondary antibody on that, and then the particles will get captured on the post only when the target protein is captured. So only when the target protein was captured in the particle will the particle get captured in the post, and now you get a different signal from the input and output as compared to the cells. So the beads will give you different signals from the cells at the same time, and you can also measure the proteins from the same assay. So we demonstrated this for IL-6 very recently, and essentially this shows the IL-6 picogram per ml. The limit of detection is about 120 picogram per ml. And here's some data from about 30 patient samples. Some were spiked plasma, about 10 or so were patient samples, the rest were spiked plasma. And we get a good correlation from this, and we're working on further refining it and improving it. So this is very exciting, and also we're working on translating this technology for measuring cells and proteins from the same exact drop of blood, from the same assay. We have a company called Prenosis, which now has Series A funding, and we're working with Foxconn to actually make a cheap reader and cartridge to be able to develop this technology. The company is also very interested in actually combining EMR of patients and using machine learning to combine all of this to build a big database and then use the point-of-care device to add to that database to be able to then stratify the patient and their state of disease. In the last few minutes, actually, I'll talk about a different project completely. This is something I've also been working on. This is more educational, so our educational moonshot, I call it. But this convergence, so clearly what's happening here today, and in general, I mean, I think the great things have happened at MIT here in terms of convergence. So this report, I hope everyone should have read it. That should be a prerequisite of coming to this workshop or this kind of meeting. And again, the pioneers, the people who led, Dr. Susan Hockfield and others are in the audience. And we also had written a perspective, some of us, about around that time, with this idea of engineering in medicine. I mean, true integration of engineering in medicine. It's something that still, there's a lot to be done. And clearly what's happening here at the Koch Institute is a prime example of that. So two things that we did on our campus, just as information. So we also started a basic science and engineering-based cancer center. We have a lot of work on our campus in the area of cancer and basic biology. So we started, and we are going in with a proposal to NCI this fall for a basic science cancer center designation, but with a strong engineering-driven. So the idea of promoting discovery and engineering for technological innovations in cancer. So we'll keep our fingers crossed and move that forward, but we have a lot of activity, and we were able to kind of pull it together with research programs that involve imaging, precision medicine technology, and big data pulled together, and then also a second program on molecular drugs, pathways, and looking at engineered tumor models. And we have a lot of educational programs that are happening. So actually, the associate director of the Koch Center is on the advisory board for this. The second thing, which is even, I think, bigger and very exciting, I've been involved in this from the very beginning. So this is clearly happening already, but what we think is missing or was missing is really sort of a more fundamental paradigm shift in the education model, especially of medical schools. So most medical school, pretty much every medical school in the world today is based on these two pillars of biological sciences and clinical sciences in the curriculum. And of course, it's all grounded with compassion and patient care. The idea of medical humanities and ethics is obviously very, very important. But the question is, why isn't engineering an equal pillar in the medical curriculum? I mean, the hypothesis is just like you can't be a physicist without knowing math. We believe in the future, it would be really hard to be a great physician, innovator, and leader without being really proficient in technology. So why not we add a third pillar of engineering, entrepreneurship, technology, innovation, and design, but within the medical curriculum, and still see if we can integrate it really well to do it in 4 years. So we think that's one of the, certainly, a pathway for a track for future medical curriculum. So this was the basic hypothesis, and it's been added for about 7 years. I think many of you know about it, and we actually are starting our first class in 2 weeks. On July 2nd, the first class of 32 students come to campus. Again, the basic idea is that pretty much most of the curriculums are based on the Flexner report from 1910 with this 2+2 model. Now, of course, all the top places have started to integrate this already, but still, the idea of having the basic sciences and then the clinical sciences where you have the required rotations and the elective rotations. This report is really interesting. You should read it if you haven't. And it's very interesting. Abraham Flexner was charged by the Carnegie Endowment, by the Carnegie Foundation to review medical schools at that time. And he was, by the way, the same person that helped start, or was the founding president of the Institute for Advanced Studies at Princeton. So he was the one that brought in a lot of Einstein and many other physicists from Europe to the US. So he was a very strong advocate of basic science. So there's this discussion earlier about basic science versus translational science. And the idea that if you're doing really good basic science, great translational will always come from that. So essentially, we looked at a lot of these models, and it's really interesting when you start from a clean slate and you don't have a medical school, how would you set it up? So we came up with this sort of a model after talking to lots of people, and actually the leaders of the HST program here were very, very helpful, gave us a lot of very good advice, and many of the places we talked to all the top people in the field. So essentially the idea of really integrating the clinical science from the very beginning, the basic science is pulled back a little bit here and more condensed. Engineering and innovation is then fully integrated into the curriculum all the way across. And of course, medical humanities as a fourth pillar is also very much integrated. So this engineering framework of how a system works, how to model the system, and how to improve the system, that's really kind of sort of permeating in all the courses, normal state, disease state, and then trying to develop as much as you can modeling in between to be able to predict and re-engineer the system to bring it to steady state, which is a healthy state. So then, imaging, nanotechnology, computational medicine, data analytics, AI, privacy security, biomaterials, devices, synthetic biology, all these sort of elements are just integrated into the curriculum with, of course, the framework of the healthcare systems engineering, looking at trying to be aware of the whole system also. So just my last slide, I think. So there's sort of 4 ways of how engineering is integrated. It's actually fully integrated in all the courses. For every organ system-based course, we have 3 course directors, a physician, a basic scientist, and an engineer who have been working on developing their curriculum. We have these idea projects from every required clerkship, capstone projects, and a data science project also that every medical student would be required to do. So it's very exciting. I think this has been a big effort with everybody from our campus involved, more or less, chancellor, provost, dean, and I've been very fortunate to be involved in it from the very beginning. So with that, I'll end. I also thank my group for the research work and all the great collaborators, and thank you for your attention.